I'm going to talk about something that you don't hear about very often, which is using Hi-C to study viruses. Hi-C basically was a method that was originally developed for looking at how a genome folds in 3 dimensions. And we have adopted it to do basically everything else except for the canonical application of the method. And so this data type is super cool because it allows you to answer 2 questions. One is it tells you which parts of the genome are close together or far away from each other, not just in 3 dimensions, but also in 2 dimensions, so on the chromosome. And if you know which parts of the genome are close or far, you can do things like scaffold a genome of any length, which is, I think, what we're sort of most known for. You can haplotype phase a genome from end to end full chromosomes. You can, it's a very robust and inexpensive way of detecting large scale structural variation, translocations, inversions, et cetera. So you can use it for cytogenetics as well. But what I'll talk to you mostly about today is the power of Hi-C to deconvolve a mixed community. So basically, Hi-C also tells you when 2 sequences start inside the same cell at the beginning of an experiment. And we've leveraged this, this property to do a lot of super neat things in the field of microbiome. And that's our newest stuff, and I'll talk to you about it today. This is sort of if you guys are interested in genome assembly and scaffolding a genome, this is something, it's a well-trodden path. We've published this hundreds of times. It's been kind of in the popular press and all that. So if you guys are interested in having a genome assembled, phased, et cetera, please reach out. But this is the key slide of the talk. Because the way Hi-C works is you take cells when they're still intact and you treat them with a cross-linker, aka formaldehyde. It will cross-link DNA molecules that are nearby each other in 3-dimensional space, this. But this cross-link happens before the cells are lysed. So if you were to have a mixed microbial community, every Hi-C junction within statistical sort of reasons, limits, every Hi-C junction hangs out inside the same cell. And so you don't get any Hi-C junctions, presumably between different cells. And so what that means is that if you have a microbial community like this, if you just do normal shotgun sequencing on it, you have these contigs that come out of the assembly. You don't know which ones belong to which organism. If there's a genome with multiple chromosomes, it's very difficult to know that all these chromosomes belong inside the same organism. If you have a plasmid or a phage, it's very difficult to know where this phage is coming from, where the plasmid's coming from. And that's what we're addressing here, because if you do have Hi-C data, what it will do is it will create physical junctions inside of all these cells. And you will have intra as well as inter-chromosomal junctions. And you will know which contigs are touching each other, and you will know which contigs belong in the same genome. And if one of those contigs happens to be a plasmid, you will know who it's interacting with, which cell it's coming from. So you can actually do not just a metagenome deconvolution, you can actually track mobile elements in vivo directly in microbial communities without having to culture things. So first of all, we have a platform called ProxiMetal that uses Hi-C data to bin metagenomes. And we've shown that it essentially takes normal metagenomic principles of binning and supercharges them with this orthogonal data type, Hi-C, that empowers you to basically extract more genomes from a microbiome sample, better genomes, more complete, more accurate, et cetera. That's what these orange dots represent from a given sample. These are all the bins, all the genomes that can only come out, only ProxiMetal would get. But beyond that, right? So mobile elements, bacteria are not the only things that live in microbiome communities. One of the key components, the most numerous component of most microbial communities is actually phages. We all know that antibiotic resistance is spreading like wildfire all over the world. And one of the reasons why it's difficult to track and why it spreads so quickly is because it bounces around the population on mobile elements like plasmids and viruses and more sort of conventional methods of horizontal gene transfer. And so just just sequencing a sample and saying we have AMR in the sample is not enough. You have to really understand sort of who the hosts are. Are there multi-drug resistant organisms that are carrying, 8 resistant plasmids in them? Phages for phage therapy, for fecal microbiota transplants. Understanding phages and who they're infecting is super important, and it's just very difficult to do with conventional sequencing methods. And what IC does, and what we've recently done, we've recently added several features to the platform. Essentially, it allows us not just to bin bacterial genomes with this data, we can also annotate AMR genes that are inside of those assemblies and connect them to their hosts. So we can figure out which AMR genes are residing inside which hosts, and also whether they're being carried by plasmids or by they're integrated in a genome, maybe sometimes even by a virus. And so we can simultaneously get really deep into this. But the newest thing was we came up with this one method that takes advantage of the following. If you have a phage somewhere in your metagenomic community, and there's 2 contigs that belong to the same phage, those 2 contigs will be touching each other. They will interact with one another, just like bacterial contigs in the metagenome assembly. So the two phage contigs will be touching each other, but they will also be touching the same host. Okay. And if you have those 2 pieces of information, you can not only take a known phage and affiliate it with a particular host, you can actually de novo reconstruct phage genomes from a metagenomic community without any culturing. So you can simultaneously actually reconstruct viral MAGs and connect them to their host at the same time. And so we published this a little while ago. We call this algorithm ProxiPhage. It takes IC data, and so here's a figure sort of demonstrating what I mean. Every pie slice in this figure represents one of these VMAGs, a viral MAG, a viral little genome. And the fact there's this line here, it means that these are contigs that are all touching each other and also interacting with the same bacterial host. And so this is a phage, right? This is a phage genome. We've shown, again, bounced it against long read assemblies where the entire phage is a single contig, and it's a surprisingly accurate way of doing this. The cool thing about it is that this is all done from shotgun Illumina data, right? This is all short read data. It can be done on long reads as well, but from short read data, you're able to pull out something like 2 to 3 times more phages than you would from long read assemblies, simply because there's just the Illumina reads are better at kind of reaching into the sort of the low abundance parts of the metagenome. And not only are we reconstructing these viral MAGs, we're simultaneously, right, connecting them to their hosts. And so we can also tell, okay, well, these are the phages. So in this figure, on the x-axis are the mobile elements, the phages, and on the y-axis, the vertical axis, these are the bacterial MAGs. So we can tell which phages are touching which bacteria, right? Who are they infecting? And you can even get things like this, like a co-infection event, where multiple phages are infecting the same bacteria. Or on the contrary, sort of a promiscuous phages, where you have one phage that's infecting multiple hosts. So you can dynamically measure the phage host range in a complex microbial community without having to culture anything, without having to do high molecular weight DNA or sort cells or anything like that. And this works on all sorts of samples. So here's some examples. Again, just looking at these things, you can see just how many of these phages we can get out of a single sample without any kind of extreme effort using this algorithm. And then we can do experiments like this. This is a time course of fecal samples during recovery from antibiotic. So day 0 is the end of an antibiotic treatment. And then as you take these fecal samples, what you're seeing is over time, the new bacteria begin to repopulate the community, right? New MAGs, new bacteria are coming in. And around the same time, new viruses are starting to come into the community, right? Meanwhile, antibiotic resistance genes and plasmids are going away. So this is what you would expect, right? This is kind of conventional shotgun metagenomics sort of analysis. But remember, because we have Hi-C data for all these samples, we can go deeper. We can actually, for every one of these samples, figure out on a contig or on a mag level, which viruses are infecting which of those bacteria. And the same thing with plasmids. One thing I didn't mention is that the same logic used to bin viruses, we can do with plasmids as well. So we can actually reconstruct plasmid mags and assign them to their hosts right in these metagenomes at the same time. And so now this experiment becomes this experiment, where you're taking a time course, you can see the mobile elements hopping around from one organism to another, in addition to kind of, popping up, disappearing, but you can actually measure the host range right out of in vivo samples. And you can do the same thing for plasmids, as well as phages. And of course, you can, we can sort of bring it all together, and we can start looking at antibiotic resistance and how it moves, from from earlier time points to later time points. In this case, right, you see basically by day 29, most plasmids are gone. In the beginning, this is E. coli that's just packed full of AMR plasmids. And so we're essentially getting a much more complete picture of the microbiome, not just taxonomically classifying the bacteria that are there, but really digging into it and being able to detect new phages, new bacteria, new eukaryotes, prokaryotes, et cetera, and profiling the antibiotic resistance host range. And this is all done without culturing, without relying on a priori information. You don't need high molecular weight DNA. You don't need special machine to buy just for this. It's all done with kits. We, of course, sell these kits. One thing that I should mention is that the analytics, all those figures that I've shown you, they come for free. We purchase the kits. We have an automated platform that's bundled in with the kits. And so you can basically get all the analysis done for free along for the ride. And that's it. This is a giant QR code that if anyone's interested in the phage binning paper, put your phone in front of the screen and it'll take you to that paper.